everyone, I am Pallavi Jain and today I would be presenting my bioinformatics project titled Deciphering Heterogeneity and Cellular Crosstalk in Non-Small Cell Lung Cancer through Single-Cell RNA Sequencing Analysis. As I said, I am Pallavi Jain and I am postgraduated from Hansraj Mahila Mahavidyalaya, Jalandhar, and my interests are oncology, bioinformatics, genomics, transcriptomics, structural biology, and molecular biology. To start with our project, let me briefly tell you about what actually lung cancer is. Lung cancer is a serious health problem. Starts with abnormal cells growing fast in the lungs, forming tumors and causing breathing issues and chest pain. It's the number one cancer killer, and sadly, in 70% of cases, it's already spread far when we find it. We mainly see two types of lung cancer. One is small cell lung cancer, which is common in heavy smokers, and other one is non-small cell lung cancer, which is more common and has different kinds. Non-small cell lung cancer is a big worry because of its high rates and poor outlook in late stages. But new treatments targeting specific parts of the cancer or boosting the immune system are showing promise and could change how we fight this disease. Non-small cell lung cancer is the most common lung cancer, making up 85% of cases. It starts when lung lining cells go wrong. There are types like squamous cell carcinoma, large cell carcinoma, and adenocarcinoma. So we are always looking for new treatments for all lung cancer stages, including non-small cell lung cancer. Technologies like single-cell RNA sequencing helps us understand the disease better as it reveals cell heterogeneity, and a great example is the insights we get from the GEO dataset GSE 148071. Single-cell RNA sequencing or scRNA-seq is a cutting-edge technique that allows us to explore gene expression patterns at the resolution of individual cells. Unlike traditional bulk RNA-seq, which averages gene expression across entire tissues, scRNA-seq provides a detailed view of cellular diversity. Researchers use various assays such as SmartSeq2, DropSeq, and 10x Genomics to capture gene expression profiles from single cells. However, scRNA-seq data can be challenging due to its sparsity and noise. Understanding these variations is crucial for accurate analysis and interpretation. Now, single-cell RNA-seq data analysis workflow. Let's walk through the workflow of single-cell RNA-seq. First, we pre-process the data, which includes quality control, normalization, gene filtering, and batch correction. Next, we reduce the dimensionality of the data using PCA. Then we move on to clustering, where we identify cell clusters using methods like k-means, hierarchical clustering, and graph-based clustering. After that, we annotate cell types by assigning biological meaning and comparing gene expression. And then we finally perform differential expression analysis to identify variations in gene expressions. This workflow helps us understand the complexity of diseases at a cellular level. Now, let's move on to detailed explanation of scRNA-seq data analysis. First one is data extraction. In data extraction process, we first search the NCBI database for GEO datasets. We then apply filters like organism, study type, sample count, and file types to narrow down our search. From these, we select a relevant dataset such as GSE 148071. We then extract the expression matrix of RNA row count from our chosen samples. In this study, we focused on 5 samples representing lung adenocarcinoma, such as P16, P20, and P32, and squamous cell carcinoma, P27 and P37. Next, we prepare our samples. We start by isolating individual cells and attaching UMIs and cell barcodes to the RNA. Then we move on to uploading our input file on scRNA. We select the file type, which is 10x Genomics, and choose the regular upload system. We upload the barcode file, gene matrix file, sequencing reads, metadata file, and finally, we group our samples into lung adenocarcinoma and lung squamous cell carcinoma categories and hit the submit button. Next, we move on to sample sequencing. After preparing the samples and uploading the data in the scRNA-seq pipeline, we start pre-processing. We start sequencing. We conduct pre-processing steps for quality control and normalization. Then we integrate the data using techniques like size cell cycle, scoring, PCA, batch correction, and surrogate anchor integration. And finally, we perform downstream analysis. This includes clustering, visualization, identifying marker genes, annotating cell types, and analyzing differential expression. First, we perform quality control or QC. In this, we remove cells with extreme gene counts or total counts from the zipped file. Next, we normalize the data using SCT transform normalization with the help of scaled counts file and transformation zipped files. And lastly, we address any technical noise and confounding factors to ensure the accuracy of our data. In terms of QC matrices, we look at unique genes detected per cell, total transcripts per cell, and the proportion of mitochondrial gene transcripts. And we also use log10 transformation for analysis. In our bar chart analysis, we see a consistent cell count in group 1, but it varies in group 2. Notably, the sample SMK 345123_2 shows a higher cell count indicating potential over-representation. Next, we use PCA or principal component analysis to reduce the dimensionality of our high-dimensional data. PCA works by finding the axes or principal components that capture the most variation in the data. We can visualize this in a PCA plot, where we scatter cells based on their PC1 and PC2 values. Cells with similar gene expression form distinct clusters. The spread along PC1 and PC2 shows differences in gene expression. And interestingly, the separation along these species can reveal differences in cell types. Next, we cluster single cells using an unsupervised algorithm. This helps us organize the cells. We focus on key dataset columns, which reveals a diverse transcriptional landscape. Importantly, it helps us identify distinct cellular populations. Next, we identify and visualize marker genes. In this, we use differential expression analysis to define our clusters. We observe significant p-values and log2 fold changes. There's variation in gene expression across clusters as shown by PCT.1 and PCT.2. Adjusted p-values highlight important genes like AGR2 and SOX4. And finally, we use UMAP visualization to show the distinct cellular populations and their transcriptional states. In the multidimensional scaling and visualization section, we used UMAP for two-dimensional data representation, which showed a distinct separation of cellular populations. Clusters numbered 0 through 15 displayed varying degrees of similarity. For cell type annotation via UMAP analysis, we observed clear segregation of cells into discrete clusters, identifying various cell types such as epithelial cells, B cells, and monocytes. This distribution reflects the complexity of the tissue microenvironment. In the differential expression analysis within clusters section, we introduce intracellular heterogeneity and differential gene expression analysis. Dot plots are presented to compare clusters between group 1 and group 2, highlighting key matrices, average expression as color intensity, and percentage expression as dot size. Observations from the dot plots reveal cluster-specific gene expression insights such as the high expression of S100A8 and S100A9 in cluster 1 of group 1, suggesting specific biological roles. In our analysis, cluster 2 shows a distinct expression profile with FN1 and MMP9 prevalent in group 1. Cluster 3 exhibits consistent high expression of MMP9 and S100A8 across nearly 100% of cells in group 1. Cluster 6 shares transcriptional commonality and cluster 1 and 3 featuring S100A8 and S100A9. Cluster 9 displays differential expression of TREM2 and CD9 in group 1, while cluster 11 has notably higher expression of FTH1, RP2D3, and S100A9. These gene patterns highlight functionally distinct cell groups, with S100A8 and S100A9 emerging as potential key markers. In the UMAP visualization and gene expression analysis, each dot represents a single cell, grouped by gene expression similarity. Color intensity indicates expression level, with deeper blue showing higher expression, while gray dots represents no or low gene expression, highlighting gene like ABRBC1, PEBP, and ERBC5. We observed their roles in cellular functions. High expression areas suggest distinct cellular subpopulations, and clusters indicate specific cell types or states such as PEBP. In the ridge plots for gene expression section, each ridge plot represents a gene's expression across different cell types, with ridge height and area indicating expression density. Color coding allows for easy comparison across genes. High variability genes like ABRBC1 and PEBB show diverse expression across cell types, while cell type specific genes like SCL and SOCS3A1 peak in specific cell types such as endothelial cells. Widespread expression suggests fundamental roles, whereas restricted expression may be cell specific, highlighting potential targets for disease related genes based on cell type expression. In the violent plots for gene expression section, each plot shows gene expression across cell types with thicker sections indicating higher expression density and vertical lines representing the expression ranges. Gene-specific insights reveal high variation for genes like ABRBC1 and PEBP, while SCL is highly expressed in a specific cell type. Widespread expression suggests fundamental roles, and comparative analysis reveals co-expression patterns. In conclusion, non-small cell lung cancer constitutes 85% of lung cancers with tumor heterogeneity, significantly impacting treatment efficacy and patient outcomes. scRNA-seq reveals cell-specific transcriptomes, aiding in the understanding of tumor heterogeneity and the microenvironment. Rapid advancements in single-cell transcriptomics ensure the workflow remains relevant over time. Data analysis insights show consistent results from subsets of the dataset, despite incomplete cell type and cluster annotation. In the marker gene identification section, unique marker genes help identify specific cell types and clusters. Research findings from scRNA-seq reveals the presence of cancer cells, immune cells, fibroblasts, and endothelial cells, highlighting the complexity of cellular crosstalk and informing potential therapies. Distinct populations of cancer and non-cancer cells are mapped, with lung adenocarcinoma and lung squamous cell carcinoma exhibiting distinct molecular features. Tailored therapies based on subtype-specific pathways are crucial for effective treatments. The GEO dataset and prognostic markers section highlights the GSE 148071 dataset, which aids in identifying novel cellular subtypes and potential prognostic markers to inform clinical strategies. The potential of scRNA-seq to enhance individualized cancer treatments is enhanced with bioinformatic tools playing a critical role in cancer research. Future direction includes integrating findings with clinical data for validation and refining scRNA-seq methodologies for clinical applicability. Thank you for your attention.